So, good afternoon, everyone. I'm Cong. I'm a PhD student from Carnegie Mellon University. I work with Carl Kingsford. Thanks for giving me this opportunity to talk my work, SQUID, which is a method for detecting transcriptomic structural variation. So let me first introduce what I mean by transcriptomic structural variations, or TSVs. A structural variation is a large-scale rearrangement in genome. However, this rearrangement does not only affect genomic sequences, but can also affect transcriptome sequences. For example, the structural variation can bring together two genes and create a fused transcript with exons from both genes. And if the fused transcript translates into protein, it will create a protein with a combination with sequences as a combination of the two original protein sequences, and it will have a change in function and abundance compared to either of the original proteins. And this is well known as fusion genes, which is the TSV that fuses sense strands of both genes. And fusion genes have been identified to be drivers of various diseases. For example, BCR-ABL fusion is a driver for breast cancer, and TL-AML1 fusion for leukemia. However, a structural variation does not necessarily affect a pair of genes. It can bring together, for example, a part of the gene with a previously non-coding region or non-transcribing region and create a fused transcripts that incorporating that previously non-transcribing sequences. And we name it non-fusion gene TSV, which says that the TSV does not fuse sense strand of two genes, but incorporate some non-transcribing regions. Non-fusion gene TSVs are much less studied compared to fusion gene TSVs, possibly due to the limited method for accurately detecting them. But still, if the fused transcript translate into protein, the protein product will have a different sequence and may also have a change in function or abundance. So we would like to detect both types of TSVs, and especially paying attention to the non-fusion gene TSVs. And so we design our method to do that. We call it SQUID. And SQUID features a high accuracy in non-fusion gene TSV detection, and it also has good accuracy in fusion gene TSV detection, which I will show in result later. And SQUID is available at this GitHub site. To give an overview of the method, after aligning the reads onto a reference genome, we convert this read alignment into a graph, where we first separate the genome into segments, and each endpoint of the segments is a node in the graph. For each read alignment, no matter whether it's concordant alignment or discordant alignment, as long as it spans more than one segment, we will create an edge for the read alignment to specify the connections between the segments that the read spans. For discordant reads, it indicates that the read alignment is actually disagreeing with the library preparation. For example, the library preparation says each parent should generate from one single chromosome, but you can split it, align the read onto like one part of the read to one chromosome and the other part onto another chromosome. Like it's a disagreement with your library, and it's called discordant read. For discordant read, this disagreement can either be due to TSVs, but it can also be due to the sequencing error or alignment error. With the assumption that sequencing error and alignment error is random, and these two error-induced discordant reads are relatively rare compared to congruent reads, we filter out the sequencing error and alignment error-induced reads or induced discordant edges by rearranging the genome segments and by linearizing them to maximize the total number of concurrent reads with respect to the newly rearranged, newly linearized sequence. And the final list of TSV is detected from the novel adjacencies after the rearrangement. That is the original discordant read that become concordant after the linearization. In this tiny example, there are 5 segments, and out of the edges, there are 3 of them are discordant connections, discordant edges marked by red. And the edge weight indicates the number of reads supporting this connection. For all of the 3 discordant edges, we can make them concordant by inverting segment C and D as a whole. And in this case, the 3 original red edges are becoming black, becoming concordant. Note that the edge with, do I have a point? Yeah, the edge with 2 as with a weight 2, it's actually a splicing edge, and it's kind of can become concordant in the sense of alternative splicing. Also, there's a newly emerged discordant edge marked by red. But since the total number of concordant read is maximized with this inversion, so we think the inversion is actually valid. And the TSVs is the new adjacency after this inversion, which is the tail of segment B connected to the tail of segment D, the head of segment C connected to the head of segment E. So we test the performance of SQUID on previously studied cancer cell lines, where there are experimentally validated structural variations. So in both panels, x-axis is specificity, y-axis is sensitivity. When restricted to fusion gene predictions, SQUID has similar results, similar sensitivity and specificity compared to the top fusion gene detection softwares. But there are only 2 methods that can output any non-fusion gene TSVs, SQUID and diffuse. And SQUID has a much higher specificity and also a higher sensitivity compared to diffuse. It's very likely that when diffuse design their algorithm, they don't pay attention to non-fusion gene, but we design our method with the notion of their non-fusion gene TSV exists, and we would like to detect them. And then we apply SQUID on 400 arbitrarily chosen TCGA samples across 4 different cancer types, bladder cancer, breast cancer, lung cancer, and prostate cancer. The panel A is the number of TSVs per sample for each cancer type, and panel B is the number of non-fusion gene TSV per sample for each cancer type. We observe in this 400 TCG samples, there are about 10 to 15% TSVs that are non-fusion gene TSV, and breast cancer has more samples containing a larger number of general TSV and a larger number of samples containing a larger number of non-fusion gene TSVs. After we separate the TSVs further into interchromosomal and intrachromosomal types and compare the number of 2 types of TSVs within fusion gene and non-fusion gene TSV categories, we observed that prostate cancer, which is this column, it has the largest number of interchromosomal TSVs for both fusion gene and non-fusion gene. And when we compare the ratio of interchromosomal and intrachromosomal TSVs between fusion and non-fusion gene, we observed that non-fusion gene actually are more likely to be intrachromosomal compared to fusion gene. And finally, we're able to identify novel non-fusion gene TSVs involving tumor suppressor genes. And then this is one example we identify from the 400 TCG sample. And there are, in 2 samples, there are 2 non-fusion gene events involving the tumor suppressor gene, ZFHX3 gene. This gene transactivate cell cycle inhibitor. And in one case, it's involved in a non-fusion gene TSV with energetic region from the same chromosome. And in the other case, it's involved in in non-fusion gene TSV with the antisense strand of another gene. And however, in both cases, only the first 3 exons of this gene are kept, and the rest of exons of this gene are totally lost in the transcripts. It's an indication that with the exon truncation, the protein sequence may also be affected, and also protein function. To conclude, the algorithm of SQUID, which is rearranging GM segments based on both concordant and discordant reads, is effective in identifying TSVs from RNA-seq data. And out of the TCGA samples we tested, we observed that non-fusion gene and fusion gene TSVs have a different inter-chromosomal preferences. And we are able to identify novel non-fusion gene TSVs that involving tumor suppressor genes. And this types of non-fusion gene TSV can potentially play a role in cancer, but further experimental validation for the study are definitely needed. But this type of TSV are not able to be detected by previous methods. Our method is published in Genome Biology, and again, this is the GitHub website for our method. I would like to thank you, all of you, for listening to the talk, and I would like to thank my advisor, Carl Kingsford, my co-worker, Ming Fu, and all the group members, and also all the funding sources, especially HITSIG for providing me with the Troll Fellowship. I'm doing a poster afterwards at A394. Thank you.
We have time for maybe a quick question or two. Any questions? Okay, great. Thank you.